This is the Master Brewers Podcast, brought to you by the Master Brewers Association of the Americas, a volunteer organization dedicated to continually improving the products and processes of our membership since 1887.
Okay, let's go!
Master Brewers brings you interviews with the industry's best and brightest in brewing science, technology, and operations. Welcome to the Master Brewers Podcast. I'm John Bryce, and here with me, as usual, is my colleague, friend, and co-host Mark Sammartino. Today, Mark and I are joined by Leo Chan. We're here recording live from the World Brewing Congress in Denver, Colorado. Welcome, Leo.
Thank you. It's great to be here.
Good. Leo, you've been actually rather prolific. You've done a lot of work, published a lot of work through the MBAA. You've presented at several district meetings that I'm aware of.
Yep.
And then here at the WBC, you've also got a poster that you're presenting.
Yes.
Why don't you give us the lowdown on your poster?
Sure. So before I start, I just want to thank my co-authors on the poster. One is Jamie Scher, and the other one is Joseph Thomas from Harpoon Brewery.
Okay.
They spend a lot of time doing some of these experiments with me, and then getting the poster made as well.
Great.
So just a big shout out to them. So the poster it's basically looking at developing a method, a viability and cell counting method for looking at flash pasteurized yeasts. So the current method that we have been using for the automated cell counter that we have is called the salometer, and we have been using a dye combination called acridine orange and propylium iodide to look at live and dead cells. And what we found is if the yeasts are flash pasteurized or heat killed, they actually absorb the dye a little differently. So what we found is you actually had to stain them and allow them to settle and react for a little bit in the slide that we put in for 5 to 10 minutes. Then the signal will be clean and corrected for live and dead. Otherwise, if you don't do that, there will be some sort of crosstalk between the green fluorescence and the red fluorescence. Then you have some of a confusion between what is live and what is dead. Okay, so that was the first part of the poster where we kind of optimize how to count the yeasts. The second part is we try to use this method for looking at shandy beer, which is one of the products that Harpoon makes. And in the beginning, we looked at the yeast samples that's directly from the beer. They should be all killed. Now, when we stained them with AOPI, we found that how come all of them are stained with the acridine orange dye, which indicates live cells, and there were no PI signal, which is there's no dead cells. It looked really weird. So what they did is they actually did some dilutions. So they did a 10 times dilution, and then after that, they saw the PI signal starts to show up, and then the AO signal starts to decrease. They did another 10 times dilution, so the final dilution was 1 to 100, and all the AO signals were gone, and the PI signal is very bright. So after looking at those data, I worked with them and said, maybe there's something in the original sample that's preventing PI from entering the cells properly. So what we did is we got another sample. So from the original sample, we actually washed the cells 3 times by centrifuging them, pelleting them, and then resuspending in water. And we did this 3 times to get rid of whatever is in the original sample. And then after that, we got very nice PI signal while all this, there were no AO signals, so indicating that there were no live cells. So that is the major part of what the poster was. And then we used that to develop a protocol for preparing the sample from collecting it, centrifuging, all the way to staining and how many samples to count to get a good representation.
Was that just specific for the shanty? Was there something in the actual product matrix that was interfering with it? Or?
We're not sure. There's actually several other salometer users that have flash pasteurizer now, and then they saw the poster and they wanted to do some testing. So I also want to work with them on seeing if this is very specific to this particular type of beer. Maybe there's some fruit remaining in the sample that's causing this, causing the non-permeable PI to not to go in.
Okay. You also use this method for viability analysis, right?
Exactly. Yeah.
Okay.
So
talk a little bit about that.
Yeah. So So the AOPI is mainly used for viability, and we started doing this maybe 6 or 7 years ago. I came in to Nexon Bioscience and started developing the protocol and the method on what buffer to use and then what stain to use to measure viability. So there are two ways that people do right now. One is just the very standard, just manual counting bright field with methylene blue. And we know that manual counting has some issues that we have to fight through, time-consuming operator dependency error or something like that. So by doing automated cell counting and fluorescence, that gets rid of some of these issues. So the second method that people usually use is just staining the cells with propylium iodide. So that stains the dead cells, stains the nucleus of the dead cells. Then in the bright field image, the software will automatically count all the cells. Then in the fluorescent image, in the red channel, it will count the dead cells, which at the end gives you total concentration, live concentration, also viability measurement. The last method, which is using AOPI, the reason why we want to use 2 fluorescent dyes is this actually started with looking at yeast in biofuel samples. As you know, a lot of biofuel in the US, they use corn mash, which is extremely messy. And you cannot use bright field to count the sample. Otherwise, you have all these debris. So we came up with a buffer system that allows the AOPI to go into the cell more readily and then fluoresce very brightly. So by doing the same thing, we were able to perform the same experiment on brewing yeast. And any products that have like residual samples, a lot of trope, or they add a lot of fruit particles in there, it becomes very complex medium, then using a 2 fluorescence method would probably be more beneficial, so that in the bright field you don't count these non-specific particles as debris. That skews your results.
Very good.
Yeah.
And for those who would like to get more detail about your standard process, not for the pasteurized yeast, but for your regular yeast counting, am I correct? They can find out more about that in the most recent issue of the Technical Quarterly. You've got an article in
Yes.
Volume 2, right?
Definitely.
Yeah.
So we have we had the pleasure of working with Mark on publishing 2 technical quarterlies. One is looking at different methods of measuring viability, as what I just explained, and the second one is looking at other physiological parameters, which is a little bit more tricky, such as glycogen content, neutral lipid, and also trailose.
Right.
That's in the yeast.
So that gives you more of an indication if you have yeast that's not quite as healthy but not dead.
Yes. Currently, we're still doing some projects trying to identify, what type of correlation is there, looking at vitality, looking at glycogen content versus how the fermentation goes.
Right.
So it's definitely a little more difficult to do this with breweries. So I am working with fermentation science departments at universities to trying to do some of these experiments. I think probably the latest collaboration was with University of Maine with Dr. Jason Bolton, where we looked at how to use the salometer and AOPI to count Brettanomyces, and that's getting very popular.
Yep.
A lot of the breweries are trying to look into using Brettanomyces in their product, but it's very hard to count.
Yes. I can imagine.
So we used AOPI to stain the yeast and be able to look at individual nucleus inside these pseudohyphae that the Brettanomyces form. So these will be later also published in the journals. So and also looking to put this in the Technical Quarterly.
That's great. And how long have you been a Master Brewers member, Leo?
I believe 6 years now.
Great.
6 to 7 years, a long time. And the funny thing is, I never had craft beer until I moved to Boston. And then, when I was in school, mostly I just, drink, lower cost beer.
Yeah.
So when you're in college, you look for those. But, I never really enjoyed it until I moved to the New England area and started working with this community. It's really fun, really very different than other communities that we work with. We're usually in the life science department. So this is a very niche, unique community that we work with.
That's great. What's been your favorite member benefit of being a Master Brewers member thus far? I mean, you've got, obviously there's the networking opportunities, there's the TQ, there's also the resources on the community site, kind of having some question and answer. What have you enjoyed the most so far?
I think one of the things is being able to just go to the MBA website and just get the technical quarterly. If I need anything, just sign as a member. It's very, very good. And a lot of nice publications are in there. Really very, very good studies.
Even very old ones going back 50, over 50 years.
Yeah, all the classic, classical ones. And the second thing that I really enjoy are the daily forums that gets emailed out. And looking at what people are doing and some of the topics that people are talking about, I think those are very useful. And you have those type of platforms so people can share information is really good.
It's a nice pulse too. You get a feel for what are the issues that are out
there.
Yeah, that are
exactly.
People crazy.
Especially from a technology development perspective, we're always trying to see what do the end users need. Right. What do they need in order to help them with their science or technology development? That's what we look for.
Very good. That's great. Well, Leo, thanks a lot for joining us. And,
thank you.
Hope you enjoy the rest of the show here, and have a great day.
Thank you.
Appreciate it. Type Chan into the search bar at mbaa.com to find Leo's articles, presentations, and posters about techniques for automated yeast cell counting. Next week in episode 8, Dave Boffman joins us to talk about boiler safety, mistakes many brewers make when purchasing steam boilers, and some pros and cons of atmospheric versus forced draft boilers. Just like that one day, like everyone else did.
The two leading causes of accidents each year are operator error, poor maintenance, which is under one category, and low water cutoffs. So it's important that training be such a high priority in this whole process, and that's probably the biggest part of the equation that's missing.